From
Yeah.
1, 2, 3, start.
Okay. Can we start now?
Yes.
Yes.
Very good evening, respected Surveen ma'am, Rajvi ma'am, Teena ma'am, Shashi ma'am, sir, and all the dear participants. Um, uh, I welcome you all to today's webinar on male infertility and the role of supplements. This session is organized by IFS SIG Holistic Medicine and SIG Endocrinology along with MP chapter. We all encounter cases of male infertility in our day-to-day practice. Male infertility accounts for around 25% cases of infertility by alone itself, like the male infertility itself accounts, and also contributes for another around 25% cases. So let's begin today's session with the blessings of our dynamic secretary of IFS, Surveen ma'am. I request ma'am to please give the welcome address.
So, good afternoon, everybody. It's a pleasure being on board. I think SIG Holistic Medicine is taking the holistic medicine from chapter to chapter and from SIG to SIG. And thank you for that. I think you're doing a wonderful job. Along with them is SIG Endocrinology and Madhya Pradesh chapter. And on a very, very important topic, which is antioxidants, a simple topic, but very, very important. And we generally see prescriptions which are just antioxidants left, right, and center. We actually want to know whom to prescribe, how long to prescribe. It is so very important, and which antioxidant to prescribe, because whoever walks into our OPD chamber says right drug, we just write it blindly. So this is one very important thing which needs to be discussed. And congratulations to Rajvi, Shalini, and of course the endocrinology as well as Priya Bhave. So without wasting time, let's go on to the academics.
Thank you so much, ma'am. Now I request Teena Gupta, ma'am, to please give the welcome address on behalf of MP chapter, as Dr. Priya Bhave, ma'am, has not joined yet.
Good afternoon, everyone. I bring greetings from Madhya Pradesh chapter of Indian Fertility Society. I welcome you all on behalf of Dr. Priya Bhave. Today, we are here to discuss the important topic of male infertility and the various antioxidants prescribed in male infertility. As rightly pointed out by Dr. Surveen ma'am, we in our OPD daily encounter many prescriptions in which antioxidants are being prescribed even to the patients when the semen parameters are normal. Is this correct? Are we heading in the right direction? And why is the male infertility increasing so rampantly? So today on this platform, we are going to discuss all these issues one by one. And I thanks Dr. Shalini ma'am especially to collaborate with our chapter as a part of this SIG holistic approach. Thank you, ma'am. So I request Dr. Devanshi to proceed ahead.
Thank you, ma'am. Now I request the convener of SIG Holistic Medicine, Dr. Rajvi Mehta, ma'am, and chairperson SIG Endocrinology, Dr. Shweta Mittal, ma'am, to give introduction to the webinar.
So, good evening, everybody. And thank you, Dr. Surveen, for getting this SIG. When we started the SIG Holistic Medicine, initially people wondered what this role of holistic medicine would be in this super specialized field. However, as time goes by, in the last couple of years, and especially the last few months, we have been getting requests from other SIGs to collaborate and do joint events with them. That just shows how one is moving from very focused approach to a holistic approach to the diagnosis and treatment of infertility per se. This particular webinar is primarily going to look at male infertility, and very specifically, we are going to look at the issue of decline in sperm counts and the generalized treatment that is available for male infertility. When we started, and I've been there in this field for a very long period of time, we always used to say female infertility was the major cause of infertility, with male 50%, about 20% would be male, 28% would be unexplained, and another 10% would be both factors. Over the years, all of us would agree that there is a tremendous rise in male infertility. I mean, in nooks and corners of the world. And what is the cause? Why is it being on this continuous rise of male infertility, and how far is it going to go? Obviously, there is something out there which is causing this drop in sperm counts and the treatment. And I always believe that male infertility is something which is given not given due importance. And I would just like to exemplify it by saying the general perspective. Society perceives infertility as a female problem. Although things are changing, there were times when the male partner would not even come for infertility diagnosis or even for semen analysis. After the female has been tested, the male partner comes in. Second thing is, whom do they approach? They'll approach a gynecologist. So that itself shows our bias towards females as compared to the males. Then as we proceed further, even when it comes to diagnosis, what do we do in the female? We will do follicular monitoring, endocrine profile, maybe a hysteroscopy, laparoscopy, the works, endocrine profile. But in the male, we do a semen analysis. And after that, what? Either you go in for some kind of concoctions, which I call it a mixture where there is not even much evidence for treatment, but we try out everything or go for NART. So I think we need to look at both the cause for this rise and a scientific plus experimental approach or experiential approach to the treatment. And that is the reason we are holding this webinar. And I'm really happy to see the attendance and to collaborate with different SIGs and special chapters. So thank you so much, Dr. Surveen, for supporting this. And I also thank my co-conveners, Dr. Shalini and Dr. VP. Over to you, Dr. Devanshi.
Thank you, ma'am. Thank you so much for throwing light on this important topic and telling us why it's so important. So proceeding with today's...
Devanshi, I'll just take a minute.
Yes,
Dr. Rajvi has very clearly highlighted, when we were given this SIG holistic medicine, we thought, what is it? What will we cover in it and what will we do? But the more we do, the more we get into it and the more we discover that there is so much to do actually. Because it's not only the traditional medicines, I mean, the allopathic medicines, but there is a lot, there is a yoga and there is a holistic medicine. Everything plays an important role. So many, we are becoming wiser day by day and they are really eye-openers. Thanks to the IFS, Dr. KD sir and Dr. Surveen for giving us this platform of SIG holistic. Yeah, you can continue, Devanshi.
Thank you, ma'am. For today's talk on declining sperm count and why, I'd like to invite our speaker, Dr. Rajvi Mehta, ma'am.
Dr. Shweta Mittal would also give the little brief about there. Dr. Shweta, yeah, you go ahead and give.
Shalini, lots of good work going on in IFS. So being convener of SIG endocrinology, it's a very, very large field, but what we really want to deal with in the SIG endocrinology is basically all the endocrine issues which are dealing with male infertility and female infertility. So anything and everything which covers that is something which my SIG wants to look into. It's a wonderful platform. Today I can see lots and lots of good lectures and a great panel. So looking forward to a great discussion. Thank you so much.
Thank you so much, ma'am. So for today's talk on declining sperm count and why, I'd like to invite a speaker, Dr. Rajvi Mehta, ma'am. Can I have a CV, please?
Devanshi, beta chairpersons को भी invite कर लेना अभी.
Yes, ma'am. Rajvi ma'am is a PhD in human IVF, has over 3 decades in human IVF experience as a researcher, clinical embryologist in training, conveyor IFS, SIG and holistic medicine, joint secretary of the Academy of Clinical Embryologists, recipient of first ESAR Young Scientist Award and champion of ESAR and President Commendation Award of ESAR, consultant at Cooper Surgicals India and Trivector Biomed. Author, Imagine If Stories of Ordinary People with Extraordinary. Welcome, ma'am. I'd also like to invite our panelist, Dr. Sunil Jindal, sir. Please share the CV of Sunil, sir. Sir is the consultant andrologist and scientific director at Jindal Hospital and Fertility Center, vice chairperson of Delhi ISAR, Delhi IAG, has been a gold medalist and has been awarded with multiple awards from different organizations. Welcome, sir. Next is Dr. Shashi Srivastav, ma'am. Ma'am is the director of Saurabh Nursing Home and SMC 10 number Market Arera Colony, president of Bhopal Menopause Society, life member of IMS, PCOS Society and Foxy. Welcome, ma'am. Dr. Shabana, ma'am. Dr. Shabana ma'am is the professor in Department of Obs and Gynae in GMC Bhopal, has presented many papers in national conferences and published many papers in journals, member of esteemed team of PPH Bundle, past Joint Secretary of Bhopal Obs Gynae Society, past Treasurer of Bhopal Obs Gynae Society, past Secretary of Bhopal Obs Gynae Society and Executive Member of MPESR. Welcome ma'am. So over to Rajvi ma'am.
The topic that I'm going to discuss is the decline in sperm counts, a prelude to the subsequent discussion on the use of supplements for male infertility. So first and foremost, I think even in the absence of data, all of us would agree from a personal experience, if you've been there in the field for a few years, that there is surely a decline in sperm counts, even if you are new to the field from what we have studied and what we see. And I'm going to highlight on the potential reasons, primarily most of it associated with the environment and lifestyle. And that is my focus, being the convener of this SIG of holistic medicine. So first and foremost, as I said in my prelude, that when we talked about infertility per se, in the 80s, in the early 90s, we always said that most of the infertile cases were due to female infertility, with about 20% or 30% being male, but generally that numbers were much lower and the rest were unexplained, or 10% with both partners being responsible. However, things have changed drastically, and 50% of our cases are male infertility and sometimes even more. The first paper which discussed about decline in sperm counts goes way back in 1992, where it was published in the BMJ, where they did a meta-analysis of 14,000, nearly 15,000 men over the period of 50 years, and reported that the mean sperm count had decreased from 113 million to 66 million. Now, once this paper came in, it not only highlighted this observation in the scientific fraternity, but it also made mass news. And the question came in immediately, that was there a real decline? Because 50 years before this paper was published, why did people do a semen analysis? In most cases, it was not for infertility or even assessing fertility. In most cases, it was due to the testing of some kind of infertility devices or pre-vasectomy. So there was a lot of, secondly, there was a lot of methodological variation. You know, when you're looking at the way semen analysis was done 50 years before 1992 and much later. So considering that, there was also a selection bias, and there was the statistical methodologies were commented, and people looked at it a little skeptically, thinking that there was no real decline in sperm counts. We were not comparing apples with apples, we were comparing something else. And that is the reason we need not be so frightened about it. But what it did, the impact was that there was so much of media coverage, it got people alerted to the fact that there could be something happening to the male. And many laboratories started looking at their own data, because to see whether what the paper reported was seen in their own lab. And it gave us a lot of information. One, there were some reports which came in, there was a genuine decline. And they also found that there was a geographical variation in sperm counts across the globe. One thing that happened was that I had been in the field, and this paper highlighted my own attention. And what one wanted to do was, it looked in the city of Bangalore where I was working. And the idea was to see whether we saw any kind of change, because subjectively we always felt, oh, counts have decreased. So we started looking at our hard data over a period of 1992 to 1996. And what was found was that in the normospermic men, also there was a decline in sperm count concentration from 69 million to 43 million, and there was an increase in the incidence of oligospermia. We tried to figure out what was it due to, and Bangalore was a growing city then. There was a lot of construction going on, which indicated an increase in air pollution. So we sort of went and got data from the pollution control labs, and we found that there was a corresponding increase every year with the suspended particulate matter, about which now we are very much aware and alert about PM 2.5. So this was one of our earliest reports. However, over the years, so, there have been reports coming from across the world, many labs in India, but I will just highlight the current world data on the decline in sperm concentration or change in sperm concentration. Reports coming in the last 4-5 years. China specifically looked at donors. So these were a good group of individuals, healthy individuals. And in a span of 11 years, they found that there was a change in sperm concentration from 62 million to 32 million in 2019 over a span of 11 years. Later, US somehow did not, in that report in World Journal of Men's Health, they did not see much of a change in sperm concentration. However, US is a large country and they had not explained or which profile of the individuals they had studied. When they looked at it in North Africa, what they found was that there was again a decline in sperm concentration over a period of 5 years in all the semen parameter study, and that is a huge group of 20,000, 21,000 samples. And various studies from Europe and a meta-analysis done showed that there was a 32% decrease in mean sperm concentration over the span of 50 years. So all in all, there is data from all parts of the world, all continents stating that there is a decline in sperm concentrations, a factual decline, which I'm sure anybody working in this field has also observed. When we look further over the trend and you look at the total motile sperm count, we can clearly see that there was a drop from 1995 to about 1999. Then there was a plateau. And then again, you can see that there has been a drop over the years. And now it has really decreased to a great extent. Even when we look in different age group of individuals, less than 30, 30 to 40, 39, and more than 40, the trend is pretty similar. And we can see that this graph is quite scary in the sense at the rate at which the sperm concentration is declining. Yes, it has not reached the levels of infertility as such, but yes, there is a concern that why is this concentration sperm counts decline? Then we look at the donors over a span of 7 years, a detailed analysis done by a Chinese group, and they are sperm donors, and sperm concentrations from 2008 of 86 million per ml had gone down to 57 million in 2014. Even if we looked at motility patterns, we look at total sperm count, there has been a decline, and these are donors. Of course, one concern, one may not be concerned that, it's not going to go down to very low levels, but my concern is why should it decline? Whether it is declining to the infertile levels or not, but what is happening? And obviously, we cannot cause any kind of genetic causes. It has something that has happened in the last 20 years, which has brought this about. And this something may be more to do with the environment. So when we look at how our lives have changed in the last 20, 30 years, when this decline has been so predominant, the first is the dietary changes. Today, we eat food from all over the world at all times of the year. So that is a big dietary change. Also, the agrarian society occupations have changed. 20 years occupations which existed 20, 25 years ago have disappeared now. And there are new occupations, and many of them are sedentary occupations. Would that be affecting? Dietary habits have surely changed. And there is increased consumption of processed foods, which is earlier, which was concerned to be a problem only of the Western or affluent societies, but now it's there all over. Big changes in the air pollution and environmental pollution and the introductions of pesticides and plastics. Plastics have invaded our lives in the last 30 years as never before. I mean, 40 years ago, people never had maximum, they had water bottles of plastic. Today, all the water that we consume has been stored in plastic tanks, whether they are the storage tanks of our buildings, or whether we buy bottled water, or we carry bottled water. So all of this and an invasion of the cell phones and tablets have altered sleep patterns. Earlier, sleep patterns were altered only people working in the nursing profession or working night shifts. But today, everybody is associated with altered sleep patterns. Now, do they really make a difference? So are we trying to bring about forceful associations, or is there a real association between our lifestyle changes and sperm? So let's look at air pollution. And as I mentioned earlier, we had our earlier study reported in 1997 from Bangalore, where we compared the sperm concentration to the pollutant levels across the year, over the years. And now we found a very strong negative correlation between sperm concentration and suspended particulate matter, which we now called as particulate matter 2.5. The question was why the reason why particulate concentration increased is well known. But in what way would it affect? So at that point of time, we really wanted to study the traffic policemen who were, assigned to highly polluted areas, and we tried to see if their sperm concentrations are lower or, in the severe oligospermic range, but we could not really manage to do a study to motivate policemen who have children to come and donate semen samples. However, much later, we have found that there are reports that had come in. So even in Athens in Greece, they found there was a significant decline in semen volume and sperm concentration over the years. And they sort of said that at the same time, there was a deterioration in air quality. We actually showed a direct correlation between air quality, specifically particulate matter and sperm count. And then there was another study which looked at toll booth workers. Now, people waiting at toll booths to collect tolls are exposed to a high amount of pollution because they're sitting for the whole day sedentarily, but more so being exposed to the traffic all the time. And here too, they found that the total motility of the sperm, forward progression, and sperm kinetics was significantly lower in these men as compared to controls living in the same area. There was also a significant lower amount of sperm concentrations, total sperm count, and motility in this group, as a higher DNA fragmentation. So what we intended to do with our policemen in Bangalore, we could not achieve or motivate people, but they did find a similar response. The question that comes in, and another meta-analysis done much more recently showed that exposure to air pollution was associated with a high impaired semen quality, declined sperm concentration, and reduced sperm count. Now, the question that comes in, if there is air pollution, one can understand an effect on the respiratory system. How does it affect sperms? How will it affect the reproductive system? Interestingly, a study was done by Yang et al., and they looked at the potential mechanism where particulate matter affected spermatogenesis. So what they did is they exposed mice to chronically to diesel exhaust, and diesel is a major contributor of particulate matter 2.5. They did find a decrease in sperm count and motility. But what was interesting about this study is that when they further went on to look at the potential mechanisms why with the sperm count decreased, they found that in these mice, the repair of, there were increased double strand breaks, and which during the meiotic division, which disrupted the spermatogenesis and affected the testicular gene expression. So you clearly had a mechanism by which this particulate matter affects sperm counts. And this goes on to show that if this is the case, with the increase in air pollution across the world, we are looking at quite a bad scenario, because if there is a break in the double strand breaks, we are going to have this increasing over the time. A second most dramatic change that we have now been exposed to is the pesticides. And we do know how pesticides often act as phytoestrogens. They are the endocrine disrupting agents. So if a woman is pregnant with a male child and exposed to pesticides, then these phytoestrogens affect the gonadotrophin production and affect the male and decrease spermatogenesis. Furthermore, with the increase in obesity, these pesticides accumulate in the adipose tissue in the fat cells, and therefore they again behave like false estrogens and affect spermatogenesis. Now, the fact is that who is exposed to pesticides? What does the evidence say? And in this particular study, they found that exposure to DDT, thanks it is banned now, or vinclozolin, there was a drastic effect on these toxic in the testis disease frequency. And if one looked at what the previous literature says, if we look at plastics, we can see that it is another major cause of testis disease frequency along with the pesticides. And so also it was found to some extent in jet fuels. Now, the impact of pesticides exposure was, unfortunately, there was a time where the pesticides were not often used in the country where they originated, where they were manufactured, but used in other countries with a low socioeconomic or low economic activity. And unfortunately, dibromochloropropanone was used in Philippines. Although it was banned in the US, it was still being used in Philippines, and nearly 26,000 males were exposed to DDCP. 64% of these men studied had azoospermia or oligospermia. So directly showing the evidence that the pesticide use had caused drastic reduction, not only in sperm count, but actually causing azoospermia. And in 1977, when this was used before it was banned in Philippines, they found that all the people who were exposed to DBCT, that is all the applicators, became azoospermic, clearly showing how harmful this pesticide is. Now, even if we don't talk of dibromochloropropanone, there are multiple pesticides which are out there in the market, and it is not just the farmers, it is not just the applicators who are affected, but anybody would be having some impact of these pesticides because they do enter the food chain. We consume the fruits and vegetables. Animals consume these plants, and therefore they would enter the meat, they would enter the milk, and we are all going to be exposed to it, maybe to different levels. And this becomes another major cause. Also, we need to bear in mind that even in urban India, pest control is very common. Often we pay for it for the whole year and we'll have somebody to come and do it. These are not always herbal pesticides. And these pesticides, if we are going to have them applied in a closed room with poor ventilation, they remain there inside, and we will need to be starting getting concerned about these pesticides. Then comes plastics. Plastics, we can have an entire webinar on plastics and the impact on reproductive health. Where are they used? They are used in the can linings. We use it in everything. Like I just said earlier, we use it all our water. Even food. When we transport food, we think that plastic will prevent leakage. We feel this is a high quality of plastic and it is not going to impair. But often if you carry a lunch box containing our vegetables or dal, which has oil, you'll find that the color stains remain on that plastic. So if they remain one way, there is also leaching of plastic into the food, and that is going to be found. When they analyze the presence of phthalates in breast milk, cord blood, urinary metabolites, phthalates have been found, which clearly indicates that plastics are causing. Now, what it says is that these mice were exposed to bisphenol A, and 2 of the 3 strains of mice. Now, in this particular condition, we need to bear in mind that we are exposing our own children to plastic water and feeding bottles. In fact, milk is often given to the child, to the toddler, in plastic bottles, in these feeding bottles. And we wonder what we are doing to the child and what we're going to do for the future of the child, because all these plastic bottles we are exposing these male children to bisphenol A, and we can imagine what's going to happen 20 years and 25 years later to their sperm count. So it's something which is seriously something we need to look at. We need to be alerted about it. Obesity, I think that's so much spoken about. The figures are astonishing. 2005, the reports for the WHO were that there were 400 million obese people, 2015, 50 million. And it is expected by 2025, just 2 years away, it will be 1 billion. This is definitely something which is scary, and we do see the impact of obesity. It's not just the male, even in the female, we see so much of PCOS associated with obesity. Why? Well, metabolism has changed. There's sedentary lifestyle, increased calorie intake, junk food, processed food. And again, this can be an entire topic for discussion for a webinar or an entire workshop. Now, what does obesity do? First and foremost, with the increase in the number of adipocytes, physically there is going to be issues, and we have the andrologist here. Increased obesity is seen with increased oligospermia, even azoospermia, increase in DNA fragmentation index, and decrease in semen volume. When we look at the hormonal profile, these adipocytes can clearly absorb these also act as secrete estrogens. The fat cells secrete estrogen, which will bring about a reduction in testosterone, increased insulin and leptin. So overall, it just messes up the endocrine system. And then finally, I would like to talk about the change in the sleeping light patterns. Now, the smartphone and tablet usage has gone. People are awake till midnight, beyond midnight sometimes. Earlier, this was only associated with people who were at jobs where they needed to work the night. But now it is much more, even without a necessity, we'll find people sleeping very late. Now, this change in sleep and light pattern, does it affect? And in a study of chronobiology, which clearly shows that there is an entire science which is looking at the biology of time, shows that increased smartphone and tablet usage in the evening, especially before bed, they have found a significant negative correlation with sperm motility, the decrease in sperm progressive motility and sperm concentration, and positively correlated with immotile sperms. The sleep duration also correlated with sperm total, and so longer the sleep duration, you had better sperm total and progressive count. However, it's too premature to say how it affects. Maybe there is an endocrine effect, but this is an observational study. We still need to see more patterns coming in, whether there is a psycho, neuro, immune, endocrine axis that is being affected. But the job as the convener of the SIT on holistic medicine, it was to show that yes, there is clear-cut evidence on the declining sperm counts. It's something we as scientists, clinicians, researchers who need to alert society about these issues that are coming in. Pollution, pesticides, plastics have invaded our lives to a great extent, and people are facing health issues because of this. But we need to realize that it is not going to be limited to our generation, but to subsequent generations. And we need to take our environment very seriously, and we need to alert society in general all over about where we are heading. So what has been our progress in terms of constructions, in terms of new vehicles, in terms of pesticides usage, and now, of course, the electronic tools that we are using, but it should not be that what is our progress leads to the downfall of our own civilization. So we had civilizations being brought due to drought, floods, earthquakes. We don't want technology to be the cause of the diminishing or fall of our civilization. So as members of the Indian Fertility Societies, I think we need to really go on a war footing to alert the general public of where we are heading. And with this background, I now would like to see how do we treat this. By the time we find the cause, we educate public, it's going to be it's a big, big job. But what do we do with patients who come with this? And often, as I said before, we don't have much to offer. Very limited offerings are there. So there are a lot of supplements which are being given. So I hand over to Devanshi and look forward to the interesting panel to follow. So thank you so much for patiently listening to me, and over.
Thank you so much, ma'am. That was a wonderful session. It gave us such an eye-opening data on the impact that the pesticides, the pollution and lifestyle changes has on male infertility. And it was much needed session, ma'am. Thank you so much. Now I request our experts, Dr. Sunil Jindal sir, Shashi Srivastav ma'am and Shabana ma'am to please share their views on this topic.
Yes, actually, air, water, and food, all the things are deteriorating today and creating havoc. Along with this, the addiction, most commonly I feel, gutka is the one of the major factor, more than the smoking and alcohol. Gutka should be banned. This is the most harming thing what I feel day-to-day practice. Another
Yes, ma'am.
thing, obesity and other laptop, all these are creating various type of hazards among the IT professionals. So should be taken care of all these things. Madam Mehta, you have explained each and everything very nicely. Thank you so much.
Thank you.
I totally agree with Shashi ma'am. And Dr. Rajvi, I congratulate you for such a nice, enlightening topic on declining sperm count. And I agree with Shashi ma'am, because addiction plays a very important role in declining sperm count, as we see in day-to-day practice, as well as use of these electronic gadgets. I'm really surprised that you include this part, because it is one of the most important points that we have to see, that how many times these electronic gadgets, they're affecting, or how it is affecting sperm count, because nowadays use of these electronic gadgets is so common, and we are encountering various patients with declining sperm count. So these points, they should be notified, and we can see that the patient should be counseled on these points also. So thank you so much, Dr. Rajvi. And anything, Dr. Sunil sir, you want to add?
I first of all, let me congratulate IFS for organizing such a beautiful program. And let me also congratulate Dr. Shalini and, of course, Dr. Rajvi, as always. Your talk was to the point and extremely scientific. But having been there, dealing every day with men, and recently we also, Vineet is there, recently we are also writing a book with Cambridge University on the 6th edition of the WHO sperm, how to analyze, what to do, what its best says. And apart from that, I am a YouTuber, so I have a lot of videos which have been viewed by millions on the lifestyle and which are very, very simplified forms of this. So if I look into all the data which is available to us today, which Dr. Rajvi has presented, about a few things I'd like to talk about. First of all, the lifestyle. We realize that alcohol consumption, lack of exercise, poor diet, smoking, stress has increased. And this increase is leading to a poor sperm count. Environment, of course, is bad with endocrine disruptors, which we all know is reducing the sperm count. Obesity. We always used to think India doesn't have obesity, but India is now becoming into a, I won't say a capital, but I would say a sub-capital of obesity available. Radiation and heat is catching us. Population is aging, and aging population once again has decreased sperm count with DNA fragmentation. Sedentary lifestyle. There used to be a day when people used to be walking in the morning, running, khet mein kaam karte the. Ab sab band ho chuka. Dietary pattern has changed where a lot of pollution is there in the food we eat. Stress and cortisol, of course, is a part of our lives. Endocrine disruptors, including the Bisleri bottles from which we have water. And of course, the ubiquitous mobile phone. All these things are something which has declined sperm count phenomenally in the last 30 years. At the moment, it is still within the limits of, I would say, fertility. The moment it goes below fertility, and probably at that time, people would realize that this is one of the ways that population is being limited by lowering the sperm count. I was reading this beautiful article of David Sinclair on aging. I mean, I don't know if you've read David Sinclair, heard about him, but he is a professor of Harvard, and he speaks about and writes about and has phenomenal papers on aging and how to reverse aging, how to stay aging. And he predicts that the age of a human being 50 years from now is going to be above 120. Well, that's the prediction which is based on a small size of population which doesn't control India, of course. But however, he says one small thing. And he says that the first thing to age in our human body, of course, is the sperm. And the way we see the decline of sperm count, I'm not sure where David Sinclair is going to take us to 120, but I hope the sperm count remains at where it is or decreases less. Thank you very much.
Thank you so much, sir. Thank you for your wonderful insights. And as the discussion gets more interesting, let's move on to our final discussion on the role of antioxidants in male infertility, evidence versus experience. So I welcome our moderators, Dr. Surveen ma'am. Can I have the CV of ma'am, please? Now, we all know Surveen ma'am. She is the director and head IVF and Reproductive Medicine Center at Max Super Specialty Hospital, and has 30 years of clinical experience, has authored 8 books, 4 of which have been published internationally, has over 80 publications in journals and books, is the program director for fellowship in reproductive medicine at Max Hospital, is the assistant editor, Fertility Science and Research Journal, is the peer reviewer for Journal Systems Biology in Reproductive Medicine, Journal of Obs and Gynae of India, and Journal of Indian Medical Association, is the Secretary General of IFS, past treasurer, past editor, web editor of IFS.
Yeah, I think that's Devanshi, thank you so much. But let's get on to some academics. Thank you.
Yes, ma'am. Now, I welcome now Shalini Chawla, ma'am. Ma'am is the principal consultant, IVF and laparoscopy with Max Hospitals, Delhi. General Secretary, Delhi Gynecology Forum from 22 to 24, executive member of IFS since 2020.
Thanks, Devanshi.
So
Yeah.
I welcome our other panelists now. Has KD sir joined? KD Nyar sir?
Yes,
Sir
he's
is
joined.
the
Hello.
welcome sir. Sir is the president of IFS, senior consultant and HOD at Akanksha IVF Center, New Delhi. Has 25 international presentations in ASRM meetings since 2006 and 22 presentations in ESHRE. I welcome sir. I welcome Dr. Shweta Mittal ma'am. Ma'am is the co-director and senior consultant, Center of IVF and Human Reproduction, Sir Gangaram Hospital. Is credited for first pregnancy from frozen thawed oocyte North India and has been recipient of many awards. Then Teena Gupta ma'am. Teena Gupta ma'am is the treasurer Asha Punj Fertility and Gynecology Center Bhopal and presently the treasurer of MP chapter of IFS. Then I too would be joining as a panelist. I am a consultant in reproductive medicine, Bansal Hospital. I work with Priya Bhave ma'am. Now Dr. Pranay Ghosh sir. Pranay sir, Pranay sir is a ESHRE certified clinical embryologist, has had special trainings from Singapore, University of Nottingham, and executive member of IFS. Then I welcome Dr. Vineet Malhotra sir. Sir is the founder and director, VNA Hospital, New Delhi, Secretary Andrology Section, Urological Society of India. Then I welcome Dr. Sujatha Ramakrishna ma'am. She's the head of embryology at Nova IVF Fertility, American Board of Bioanalyst Certified Embryology Lab Director. I welcome all our seniors. So over to Surveen ma'am.
Yeah, Dr. Surveen will start, and I will just share the screen because the presentation.
So,
is the screen visible? Are my slides visible? I mean,
yeah.
Yes, it is.
Yeah.
Welcome everybody. Like I said, I think Shalini and I are speaking on a very, very important topic, antioxidants. And we have a great group of experts to answer the questions which we are going to put forward to them. I think Teena, they have already been introduced. Shalini, can you go ahead? Next slide. Yeah. So the next slide, I think we've introduced everybody who's on board.
Yes ma'am.
So we all speak about oxidative stress, and we really want to know what oxidative stress is. And does it actually impact the sperm, or is there any other factor like just a common deficiency which is impacting the sperm? So let's first understand what oxidative stress is. So Teena, what is oxidative stress? And can it have an impact on the sperm? And if it can, then what is the impact?
Oxidative stress is basically an imbalance between the reactive oxygen species free radicals and the scavenging antioxidants. Whenever there is an imbalance between these two and the quantity of free radicals increases, then this causes oxidative stress. These free radicals have basically the free oxygen molecule, which has the capacity to cause the peroxidation of the lipids in the membranes. It causes it causes damage at the cellular level and the subcellular level. And as far as sperm is concerned, it causes damage to the membranes, the DNA, and the mitochondria. Therefore, it affects the sperm motility. It also causes alterations in the sperm morphology. It increases the DNA fragmentation. It causes infertility. It might be a cause of increased miscarriages in the couples, and it may also cause the increased chances of anomalies in the future offsprings.
Yeah, very true. And basically, there are two cells which are, responsible for this. One is the leukocytes if they are excessive, and second is the immature sperm. So those are the local factors which can lead to a higher reactive oxygen species, which may cause a damage, and it may cause a genotypic and mutagenic damage. We often see that the sperm DNA damage is present in the male when we test it, and we test it because it manifests as decreased motility or abnormal sperm. So what are the causes? Next, Shalini. What are the causative factors and why is it on the rise? Divyanshi?
And the causative factors for increased oxidative stress can be classified as exogenous or endogenous factors. And the exogenous factors include smoking, alcohol, recreational drug use, pollution, pesticides, poor nutrition, temperature. All this has been beautifully covered by Rajvi ma'am. And other endogenous factors include autoimmune diseases, obesity, chronic diseases, infection. So all these factors lead to increased oxidative stress.
Another very important factor is a prolonged stasis of the sperm in the epididymis. If the male has not ejaculated for a long time, then you do the analysis, there could be a DNA damage, because epididymis is the place where, you know, when it is stored, it can get damaged. It's not the testes which causes it. And any form of immature sperm, like I had said, it will lead to a increase in ROS. And we do see that infertile men have double the amount of ROS compared to fertile men. That has been documented very much so. And things like varicocele itself, as you mentioned, it's a local cause. Varicocele is going to cause a increase in local temperature, which is going to lead to a increase in ROS. Even environmental factors like UV radiations, etc., would cause. Next. Now, how do you measure oxidative stress? Hi, Sujatha.
Hi, Dr. Surveen. Thank you very much for this opportunity. So, oxidative stress, actually, you know, there are no direct way to measure the oxidative stress. Okay, there are multiple methods which actually will help you to identify the byproducts of oxidative stress. Okay, like say, for example, the simplest one is the thiobarbituric acid assay. But now there are recent studies, they say that there is one technique which is, it should measure the oxidative reductive potential in the cells, where it just looks at the ratio of the free radicals or the oxidants and the antioxidants, which would actually tell us what is the capacity of that cell to withstand an oxidative stress assault. So that is a new method which has been proposed, which actually is found to be more accurate in measuring oxidative stress. And then it is also found to be inversely proportional to the sperm characteristics. But the other indirect methods are, you know, you look at DNA fragmentation.
Can I interrupt you, Sujatha? Is this method available in India now?
Yeah, I think it is, you know, still in the I wouldn't say the research this thing, because this publication came some time ago, but I don't think it is commercially available in India. I have not even heard of it. But then I think this, I think, would be the future, you know, to accurately measure this oxidative stress, because, you know, otherwise you are just indirectly looking at so many things. Say, for example, DNA fragmentation. This is a byproduct of an oxidative stress. And so you're trying to measure DNA fragmentation. So the direct method is this.
Can I just make a comment there, Dr. Vineet here? Yeah, so I've used Myoxis several years back. Myoxis is the redox potential measurement assay, as mentioned by Dr. Sujatha. It has been available in the country for some time. The problem was that the kit that they sent, so it's a strip test. It's like a glucose strip test. And this strip was extremely expensive. And after a while, it would lose its accuracy. So, you know, if you're going to do a test now, let's consider that you're going to measure the ROS of a person. It only makes sense that you're going to measure it again after intervention, then assess whether your intervention has resulted in a change, and whether that change then resulted in an outcome which changed. So several tests, you know, it doesn't involve just one time test of this person. It's a dynamic thing. You're going to measure it at base point. You're going to put in an intervention. Then you're going to assess what change in terms of redox potential you got, and then thereafter outcomes, whether they changed or not. And because you had this complexity of cost to the strip and the test, it somehow did not become popular in this country. So I probably brought it in first when we were doing this back in 2000, 2018, when I2 Sciences was the company that was managing this from the US. They're the ones that actually made the Myoxis machine. They've now sold it to some Swiss competitor, but the Indian counterparts that actually tried to bring it in could not sustain it. So we don't have Myoxis any more in the country, and you're back to the other tests which are based more on chemilucents and other types of tests that you would do for ROS potential.
Dr. Vineet, you said you bought this in 2018. So just a rough idea about the cost. Are you aware of the cost?
I bought the machine for 4 lakhs, and these strips we used to get for about 2,000 rupees a strip at cost after a lot of this thing. And then we had to price it, because of all the costs that you get in terms of putting this test up, we would have to price it at 5,000 to 6,000 per test.
And is it being done easily in the Western countries?
So there were 2 places which it became very popular in, and most of the studies that you see of Myoxis either come out of Hamad Medical Center in Qatar or Japan, where they did a lot of testing with the Myoxis. In the US, within the US itself, at the time, till my last knowledge, they were not FDA approved to do the test. So it wasn't available as a test off the counter to be advised to someone.
Okay, thank you.
So I would just like to add one more point here, because the key word here, which is mentioned by Dr. Vineet, is the dynamic nature of the issue. And because of that, the relevance to the outcome. See, when we measure oxidative stress, it might help actually in natural conception, because in all the other assisted reproductive treatments, the sperm is processed to a great extent. The oxidative stress is kind of, reduced because of the processing methods. And because of that, you cannot directly correlate it to the outcome. And this is actually, there is no motivation to try anything more or to measure reactive oxygen species, because the whole thing is subjected to change, and the actual sample which we are going to use for the procedure goes through the separation process.
So just to add to this, this was the simplest test that you could do. You required only 0.3 ml of sample. You didn't require to look at whether it was fresh or frozen. You could use both, and you just had to put it like a glucose strip onto this auto analyzer kind of a machine, where you would put it in and it would give you the redox potential in 2 minutes. So a very good test otherwise.
Dr. Sujatha, are you using any of these tests for the sperm DNA fragmentation?
No, we use the sperm chromatin dispersion test, because that is the one which can be easily be done in the laboratory. So we use only the sperm chromatin dispersion test, because like a tunnel assay, it would require expensive equipment. Similarly, the sperm chromatid structure assay also would require, all these would require fluorescent microscopy and incorporation of dyes, which may not be routinely be possible in an IVF laboratory. So we do the SCD.
KD sir, your opinion?
No, yes, we are also doing this halo test in our lab here. But what I was going to ask, Vineet, you see, I still remember when this myelosis kit was launched in Delhi. I was also there a few years back. And thereafter, you say it went out of fashion. What was it generally one cost of one test? What was the cost?
Sir, between 5,000 and 6,000 rupees.
5,000 to 6,000 rupees.
Yeah, he said the machine was very expensive. It was 4 lakhs, the machine to analyze.
Yeah.
One strip was 5,000 rupees.
No, sir. One strip was about 2,000. The test cost was 5,000 to 6,000.
Okay.
The machine cost you had to absorb into the test this thing, as happens with all lab equipment. You have to
when we do this halo test also, we are charging, I think, 5,000 or so for the DA5. Yeah. So which is more accurate, Vineet, according to you? Supposing you have a choice between halo and that test, which one is more accurate?
So I would say objectively the redox test more accurate, no doubt about it, because of SCD having a huge subjective variation. But I would say if you take out the subjective variability, which means if you have the same person in your lab analyzing the SCD every time, you can make that objective also. Otherwise, if you compare the two, one is an objective test, one is a subjective test.
Okay. We are
Pranay,
performing
your take on
SCSA
this
test.
test?
We are doing SCSA test, and we feel it's a better qualitative test than STD. And
Okay,
you
and
get
how much-
less amount of DNA fragmentation, and maybe it reflects better. Because as Dr. Vineet said, STD is very subjective. Calculating that halo can differ from lab to lab.
So what are you doing, Shweta? You're doing the
SCSA, SCSA test.
but that also requires quite a bit of paraphernalia, na?
Yes, you have to freeze the neat sample because this is not done in Delhi. It is transported, and the cost of the test comes to around 8,000. But
Yeah,
very
but you know, freezing in itself causes DNA damage.
Yes, freezing
Freezing
can itself have. So
Yes.
we just in our last class discussed a very beautiful, extensive article, and they said the only way to remove all these damages is simple semen preparation, whether it is swim up or it is double density. And then we came to a conclusion that possibly that works the best.
Yeah, but no, the fact that you're freezing it and then sending it for analysis means you are
It's a snap
iatrogenically also
freeze.
increasing
Yeah.
the DNA damage. And so it
And the
wouldn't be very
report which
accurate.
comes, so the report what is coming right now, it's not very high. That's what I'm trying to say in the selected
Is
group.
not high. But
No,
have you
it's-
done a comparative analysis with the halo
That's
test?
the reason. That's the
Huh?
reason. You won't get that data.
Yeah, so if you do a comparative, at least you know whether the freezing is causing it or not.
So,
Like if it is higher than the halo test,
No, it is
then
not higher. I'm saying it's lower than the halo test.
no. Okay.
It's not higher. It is when I read the reports of Delhi labs which are reporting SCD, I find the fragmentation way higher than the SCSA results what we get.
Okay.
the thresholds?
And how long do they take for the report and etc.?
Excuse me?
How long do they take to
So they
report
take
back?
about, yeah, it takes about a week or sometimes 15 days for the reports to come.
And 8,000 is what you said.
Yes,
Is
yes.
Are the thresholds same for the SCD and the SCSA?
What are you asking, Dr. Pranay?
Thresholds.
Thresholds for labeling a patient.
Threshold, I think I just discussed that. I think 25%. So it's this variation is from kit to kit and lab to lab. Some will say 20%, some 25%.
Sujatha, I just want you to comment on these two techniques. Can you enlighten us as an embryologist, which one would be better?
See, from practical point of view, SED is better. The reason being, one, you can perform it in your own lab. You don't have to like it does not involve freezing and then sending the sample to somewhere else. Second thing is, like suppose if you want to look at DNA fragmentation of the sample, which we are going to use for any procedure, say for example, today's ICSI or today's IUI. And if we have to test the DNA fragmentation, I think SED would be, is practically possible. So based on these two, like I prefer SED.
So is Nova using SCD?
Yes.
Because, you know, I've done the Comet and all, it's very complicated. It requires a whole, you know, machine and a lot of stuff. I've done it in Manipal and it takes a lot of that agarose gel and you make the slides and it's not possible in clinical practice to do it.
Absolutely. And then Dr. Surveen, if you're talking about subjectivity, okay, the sperm motility assay and then sperm morphology testing itself is very subjective. Okay, that we say is the cornerstone of infertility, workup. Even that is subjective. So, you know, like the same thing extends for DNA fragmentation also. But the only advantage is like you can do it in your own system. And if you have a trained person, at least that, that interperson variability would be minimized.
Okay, going on to, you know, Pranay, we've been discussing this, but what are the precautions you take in the lab to avoid the oxidative stress? Like it could be lab induced also, no?
So again, I'd like to thank you all for having me here. When we talk about the stresses, I think as an embryologist, I would want to say that what we want to do is not want to confer upon any more stresses on the gummies that we have been given. And our job is to make sure that the gummies that we have, we don't, if we are working with something like the endogenous stressors, which you mentioned, like immature spermatozoa, or even the oocyte, which has during oogenesis or ovulation, pre-ovulation stages, being subjected to various stressors. Our job as an embryologist is to not confer about any exogenous stresses. So these exogenous stresses can be in the form of how you handle your gametes, the time exposure of your gametes, whether it is the spermatozoa, especially the spermatozoa, because the spermatozoa, being the cellular structure that they have been conferred upon, have the lowest cytoplasmic content. So their antioxidant potential, the capability of their cytoplasm to fight the stressors is a lot lesser. Also, they have a lot of polyunsaturated fatty acid content. So they are very susceptible to the oxidative stresses. So foremost, I think we would want to optimize the laboratory environment. It is very akin to how we would want to troubleshoot an embryo culture system, in the sense that we'll talk about the temperature that the gametes are being handled at, the pH, the osmolarity, even the humidity, the culture media, the oil, and because some of these exogenous things like the culture media itself or the oil that we are using, they themselves can impart oxidative stress, and especially it's true for the embryo culture media oil. There's a lot of batch-to-batch variability. The way the media has, the oil has been stored, whether it is stored at 2 to 8 degrees away from the sunlight, how long it has been stored prior to its use. So these are small things that creep into the embryo culture system, which can confer upon the oxidative stress. Then we are having in vogue some culture media which are having additives in the form of antioxidants. These are new. Not all of us are well versed with it. But when we are using these, we should be mindful of the fact that first, they are not just empirical, they are evidence-based, because if we tell the balance of this oxidation, the oxidative stress, and give a lot of antioxidants, we may till the balance to something what is called as reductive stress. We need these the moieties like hydroxyline and peroxidase and superoxidants. These are physiological moieties which carry on some physiological processes like sperm capacitation and the sperm lipid, maintaining the permeability of the sperm plasma membrane, and further on events which help the sperm to penetrate the zona. If you're tilting that, we are imparting an iatrogenic stress to the whole mechanism. So we should be mindful not to overload the male partner with either giving a lot of antioxidants or having antioxidant rich culture media. Apart from this, I would specifically want to focus on the way we handle our spermatozoa during the ART process. We should be very mindful that there are no delays in the processing, and we should be, in fact, getting the male partner to produce the sample close to the time where we are actually wanting to inseminate or inject the oocytes. There should be no delay. So we are wanting that, for example, a pickup that has happened at 10 o'clock in the morning. We really don't want the semen sample to be coming at 10, getting processed, and waiting for 3 to 4 hours, because however you process, whether it is swim up or density gradient, I'm making point about the sperm processing, because either of the conventional methods, whether it is swim up or the density gradient, owing to the incubation period of swim up or the rotations of the centrifuge in the density gradient, you are generating again oxidative stress, which is iatrogenic. So you need to look at perhaps the newer sperm processing methods, especially for patients who have established high sperm DNA fragmentation. And I think male infertility, oxidative stress, and sperm DNA fragmentation is something that I feel is very close to the STDs, wherein getting the whole thing tested and then starting a treatment for it is more expensive than doing the starting an empirical treatment, like starting doxycycline for STDs. So here, if you just work in the prescribed manner, respecting the timelines, not having delays, and just doing perhaps take 2 samples at close intervals, working with the second ejaculate, and have a sperm sorting method which is perhaps superior to our conventional methods, we can perhaps avoid extensive testing, retesting, 3 to 6 months of antioxidants, which again,
do you routinely, do you routinely do, do you routinely give antibiotics to all the patients going for IVF?
No, that was just a corollary that I was making, that it is just like perhaps you would not want to get the WHO protocol for testing for STDs. For some of the STDs, that says that you would presume it is presumptive and you can start on doxy for 14 days. So something akin that I was trying to make a point, that getting tested for oxidative stress, sperm DNA fragmentation, putting the male partner through 3 to 6 months of empirical treatment is perhaps more time consuming rather than doing what you can using the smaller abstinence period, working with the sample which you've processed meticulously, and perhaps using XC, something that again should not be used for 100% of our patients, but especially if you have documented or presumptive higher sperm DNA fragmentation, you can perhaps resort to XC, because now we know that we are not bombarding the oocyte with the conventional insemination where thousands of spermatozoa are coming, and that the oocyte is itself endowed with a self-repair and the repair of the defective paternal gamete. So you can use that. So these are some things that we can be mindful about how we work in the lab. Again, cryopreservation is a whole different gamut of insults that imparts reactive oxidative stress to either oocytes or the sperm.
Basically, I think that in nutshell, these points which we have highlighted,
Shalini,
basically it will
Dr.
cover.
Shalini,
Yeah.
I just would like to add only one sentence to whatever has been said so far.
Yeah.
See, an important thing is prolonged incubation of prepared spermatozoa at 37 degrees Celsius is to be avoided.
Right.
Because that actually increases, reactive oxygen species and then
Yeah.
increases DNA fragmentation.
Yeah, so that is a very practical point, Dr. Sujatha and Dr. Pranay, that when we do IUI, we try to do, when the sample is processed, we try to do it as early as possible. So maybe that is the reason that we try to avoid the ROS, right?
Yes.
Because once the sample is processed, we don't want it to be kept in the incubators for long.
Yes.
So basically,
Pranay
these are the main,
Pranay had made a very interesting comment on the second sample. I want to know everybody's views on second sample as far as this is concerned. And are we using it regularly in clinical practice, the second sample? Sujatha?
Actually, Dr. Surveen, we had done a study long time ago, which was even presented in ESHRI in Portugal. So what we did was we collected the first sample, okay, and then did DNA fragmentation on that. Then we collected the second sample, which was also subjected to DNA fragmentation assay. And then the samples were processed by density gradient centrifugation and by microfluidic method. Then, you know that I forget the name of the Qualis device was there. Okay, so we checked in all this. I think close to some 30 samples we tried, like 4 times running DNA fragmentation. And we found that the second sample had significantly lower DNA fragmentation. Okay, that is one. Another interesting observation we made was, even with the density gradient centrifugation, post-processing, like there is a significant reduction of DNA fragmentation. In fact, when you compare that with microfluidics, like it went down even further. But only thing is, with microfluidics, we could recover only very few sperm compared to density gradient centrifugation. So the inference we made from that study was, getting a second sample is a much cheaper method if you want to have a good semen quality, and putting it through density gradient centrifugation would actually reduce DNA fragmentation to a significant level.
Yeah, that is just what I wanted, because we also did a study. Only thing is that we didn't do a pre and post DNA because of the cost to the patient. But any sample which was reflective that yes, it may be DNA damaged because of motility and morphology, we took a second sample. And our clinical outcome was blastocyst rate, and which was much higher in a second sample. So we are very routinely doing second samples in our IVF lab. It's even with IUIs, we have second samples.
Dr. Surveen, I like to ask only one thing. How easy it is for a husband to give a second sample?
So they are very resistant. They are very resistant, and some cannot give it. So we don't insist. We say that if you can give it, it will be better.
asking for a second sample, I think hardly 30% of husband can produce it. 70%
No, sir, you're wrong. Your statistics are wrong. We are doing it. About 70% will give, 30% will say we can't give. And we just tell them that it's not necessary, but if you give it, it'll be better. And we are even taking it for IUI samples, and it's a much better sample. Especially when we take it only when the first sample is not good. We don't take it like a routine. If the first sample is not good, we take it.
We are doing it with the patient being informed, and we are mentally prepared for it. So we are doing it for only documented high sperm DNA fragmentation. And these are the patients here wherein we are giving them adequate time. It's not that we are sending them or rushing them off within half an hour or an hour. We ask the first sample, and with the intent that we are not going to be using it, but we have it. And we are going to allow 3 to 4 hours at the time during which we have done the pickup and the oocytes are resting for the incubation. And then after 3 to 4 hours, we'll definitely ask them, and they are mentally prepared that they are to give a second sample, because we have already discussed with them that the alternate options are 3 to 6 months of antioxidants or even SSR. And they are very accepting to the fact that something as simple as a second sample will, if it does the job, they are up for it. So we are allowing more gap. If we ask them for a second sample in half an hour or an hour, most of them will not go, not give a sample, be able to give a sample.
Yeah, I just had one question before we go on to this very interesting case Shalini is going to discuss. Sujatha, we were talking about sperm DNA analysis. Now, when we publish a paper, what is an acceptable technique? Is it hello sperm is not an acceptable technique in the West if we publish it in an index journal? What is your comment on that?
See, I understand that, but the thing is, like here we are, publication comes as the next this thing, right? Like currently our intention is to identify what could be the reason. Say, for example, if it is a failed IVF or it's a miscarriage or no good embryos, to find out a reason.
That
and then
I understand.
the patient.
For publication, what would be better? Would the other technique be better, or would Halo be better?
Other techniques
what is
it? Because like that is more accurate, right? Like the other technique would be more better. But the thing is, like we are not.
I understand the practical aspect.
Yes, we are not in the research setting, right? Like we are in a clinical setting where I think more than anything else, we have to find reasons for something which has not happened. So I think if that is the intention, that we can do only Halo. But I understand that for publications, it's difficult.
Yeah. I just thought maybe you'll have an alternative. Okay. Shalini, over to you. Shalini?
Yeah, sorry, I was muted. Okay, so moving on. Now we have this interesting case which we see every day in our OPD. A couple, wife 30 years, husband 34 years, married for 3 years and is keen for conception. The female parameters, AMH, tubal testing, everything is being normal. The semen analysis, it shows 50 million per ml with 10% motility and 1% normal morphology with the strict criteria. So Dr. Shweta, question to you, what do you do in such clinical scenarios? And do you routinely advise DFI to all such patients, or you have some selective criteria, or would you prefer something else before going on to DFI?
Okay, so if the first semen parameter is this, number one, I want to see, is it a normal volume semen or not? If it's a normal volume semen and there has been no collection problem, number one, I would repeat one more sample. So 2 samples showing similar reports. So this is like an isolated, I would say an asthenospermia with teratozospermia. So 2 reports similar. Then the first my step would be to identify the cause. That is most important. So you take the history, you look at his normal habits, you look at his occupation, you look at any history of repeated genital tract infections or urinary tract infections, diabetes, hypertension, any cancer histories, varicoceles, hydroceles, hernias. So all that you have to rule out. If you can find the cause, then you treat the cause first. That is how I would believe in it. And DFI for a normal routine couple doesn't come in my armamentarium very soon. It just doesn't come. It comes pretty later if there are absolutely no causes, or if I identify a cause and I think I need to take them up for IVF. So my indications to do it would be a grossly obese male, a diabetic male, or very severely hypertensive, any cancer, radiotherapy, chemotherapy, not getting so frequently varicose. If these things are there, I would ask a DFI. See here, this patient is not going for IVF, I think has come primarily to you. But if this patient was moving on to IVF, then the approach could have been really different. But even for IVF, DFI normally doesn't come so soon.
True. So I guess the take-home message is that it is not that easy to jump to getting DFI done for every patient, and we have to be very selective. First, try to rule out the cause. That what is the cause for such a low sperm count? Dr. Shweta has very clearly highlighted all those factors, and maybe so. Dr. Shweta, as a routine, suppose this patient was for IVF, maybe tries something that natural. You given antioxidants, and you have given maybe IUI you've done, this patient is moving on to IVF. So do you mean that such patients, before going on for IVF, do you recommend DFI in those cases when the patient is for IVF?
At the moment, I'm recommending DFI to 2-3 sectors. One is very poor fertilization with good oocyte morphology, where I think sperm is the factor. Number 2 is really fragmented embryos, but see, the job of sperm is to fertilize the egg. We must understand. The second place where I'm doing DFI now is if there are 2 unexplained, very, very early miscarriages. That is the place where, DFI I am recommending. And my third sector could be, which I just mentioned, grossly obese and very severely diabetic and smokers, heavy smokers. These are the groups where I would possibly do. Otherwise, I don't. Because we personally believe that a good semen preparation technique elevates most of the problems. And most of the sperm selection techniques, what we mentioned, whether it is EXE or whether it is microfluidic, doesn't apply to very severely oligoasthenospermic sample, because either it doesn't bind to the bead or you don't have the yield, how Sujatha said. So the problem comes is when we are dealing with severe oats, how do we select the sperm?
So can I make a comment there, please? So, I would add a situation of clinical varicocele if I'm considering intervention versus a assisted reproductive method. So if I am not certain that the varicocele would make a indicative choice here, which means I either have a varicocele which is not significant grade or a recurrent varicocele where it's not been sorted, I would look at the DFI there. So the intervention has got to see most of the times, otherwise putting I think Pranay mentioned there, putting an antioxidant, advising to stop smoking, lose weight and all of that. You don't need a DFI to tell you that lifestyle factors and putting antioxidants need a DFI estimation before. That's going to be done in everyone. But you're not going to do surgery in everyone. And you may not do the third. And you may not do the third cycle of IVF in everyone, which is why DFI there then makes sense, as mentioned by Dr. Shweta.
Yeah, so Dr. Shweta, Dr. Vineet, and Dr. KD sir to give the final comment. So, do you routinely put all these patients on antioxidants, or what is your take? Because these days, I guess every one of us sees a lot of prescriptions coming with antioxidants. So, does a patient coming in the first visit to you, right away you put the patient on antioxidants? DFI, we have clearly sorted this out in this panel that we have to be selective and not jump to DFI so easily. But what about the antioxidants?
So I can tell that I only give 10% of my male patients, whether going, other than IVF, hardly any, and in IVF, not more than 10%.
Right. KD sir, what's your take on that? What is your percentage?
With DFI, we are hardly doing for 10% of the patient, not for everybody.
Right.
The thing is, this patient has low motility, low morphology. We have to look for the other causes which is causing this thing. This is not necessary to be reflected by doing DFI. So you have to look for the causes and try to treat, like mention, losing weight, stop smoking, the lifestyle factor, environmental factor. It is a long way, but they are important for such
Right.
patient.
Right. Thank you, sir. Okay, thanks, Dr. Shweta. So, coming to the nutshell, we feel that a patient coming through you with severe OATS or with deranged semen parameters, please don't jump on to doing DFIs. First assess and then take a call. Now, next coming to Teena. Teena, such patient comes to you like, obviously we tend to give antioxidants with a very, every couple do ask you this question, doctor, what should I be taking in my diet so as to improve my semen count? So is there some particular diet that you advise, Teena, to these patients?
Yes, surely diet plays a very important role in creating a high antioxidant environment in the body. So these patients should be advised to take a diet which is rich in natural antioxidants like diet rich in vitamin E, C, and lycopenes. This is basically seen in all citrus fruits, berries, nuts. The patient should have a total balanced diet containing the just proper amount of carbohydrates, fats, proteins, and fruits and vegetables. The protein intake should also not be on the very higher side, because if the protein intake is high, then it causes alterations in the testosterone levels that also affects the spermatogenesis. And moreover, the origin of the protein should be the lean protein origin, that is from the legumes and the poultries or the fish, but not the red meat, because it causes decrease in the sperm functions, the high dose of proteins. And the fats, the saturated fatty acid diet should be avoided, and the unsaturated fatty acid diet and the omega-3 fatty acid diets should be encouraged. These are seen in fish, walnuts, nuts, and flax seeds. The caffeine and the addictions like caffeine, alcohol and tobacco should be discouraged. The intake of tea and coffee should also be limited. And the vitamins like folic acid and vitamin B12 rich diet should be encouraged, which is seen in green leafy vegetables and legumes. And take the proper sleep, exercise daily, do yoga, and be stress-free. This is the advice which we give besides the antioxidants in our preparations.
Yeah, very nicely highlighted, Teena. So just on this thing left side of the screen, I have highlighted like apples, plums, bananas, tomatoes, onion, broccoli, and then seafood, poultry, whole grains, fruits, and vegetables. So these are the kind of foods that we normally avoid because we want the antioxidants to be given maybe in the natural form so that they counteract the ROS. Now, I will just very briefly go through these products because somehow they have been in the controversy. Like soy foods, many patients they come. So it has been said that they have been asked to avoid soy food. No, it is not, because there is no study in the humans that soy food do any harm. Then dairy products, Teena very well highlighted. So full fat, saturated, we should avoid. But however, the low fat and skimmed milk we can take. Now coming to the commercial milk is usually made from the pregnant cows. And these pregnant cows, they usually have estrogens. And these estrogens, they decrease the testosterone level and they decrease in the sperm production. And meat and the processed meat we do tell to the patients that okay, you're non-vegetarian, so your protein component will be taken care of. It is not always so. Meat and proteins, though they rich in the proteins, but they also contain the xenobiotics and the anabolic steroids, and which they have harmful effect on the testosterone and the semen production. So basically, we have to avoid the saturated fatty acids. And again, the processed foods like the bakery products, fast foods and snacks. These days, there's so much, like they've kind of become a fashion to eat those processed foods. Again, these processed foods, they decrease the serum testosterone level. They arrest the spermatogenesis. And it has been shown, more of these studies about the processed food, that they decrease the testosterone level more in rodents. However, in humans, it is yet to be. Coffee, tea, and alcohol to be in moderate amount, not to be in daily amount. Like alcohol, earlier it was said that you can take alcohol, but yes, not daily. It is to be restricted. Again, the sweets and the sugars, these sweeteners, they do increase the weight gain and obesity, and again, insulin resistance, and hence the increased oxidative stress. So, Dr. Shweta, coming back to you, what is your choice of antioxidants and for how long do you give it?
Antioxidants, normally in the order of my choice, I can tell you, there is no antioxidant which is superior to each other, but very simple ones can be a combination of vitamin C and vitamin E. Vitamin C is water soluble and vitamin E is lipid soluble. The other ones, so if I have to use vitamin C, I will use it in a dose of 500 milligrams or 1000. If I have to use vitamin E, I use 400 to 600. My other favorite molecule is also CoQ enzyme, but I don't mix and match. If I'm only giving CoQ enzyme, then I give equivalent to 300 milligrams of CoQ enzyme. And L-carnitine is something which I give if the motility is less. But if you go into the literature, you will see that there is no specific superiority of one over the other. Other than that, it could be selenium, zinc, and some other antioxidants, lycopene.
Yeah. So, anybody else who has a preference for antioxidants? Sir, what is your take on it? KD sir?
I advise them to take fresh fruits rather than taking this tablet. You see, this tablet antioxidant, they come at a cost. Average is around 60 rupees to 100 rupees per day. And average. We are not giving them vitamin C and E. Like Shweta mentioned, yes, in a low economic setup, probably we can only dispense vitamin C and vitamin E, which are cheaper. But
very right,
being IBS specialist, we always like to give a combination of antioxidant which costs around 60 to 100 rupees. I prefer that instead of spending 100 rupees per day on the tablet, they can spend 100 rupees on fresh fruits, which we have mentioned. And the effect is the same. Because you cannot quantify the effect of all this antioxidant which we are giving. That is my personal perception, you see. But when I also advise them some sort of antioxidant, and if you say the patient cannot afford that amount, that expenses, we just add the vitamin C, one tablet every day. That is all. It's the cheapest.
Yeah, so very rightly said, sir. It is very, very important not to, I mean, I'm not saying I'm against any of these commercial, but if you see all these commercial preparations, they have somebody, each company people would come, ki, okay, now our product is better. The other company person would say that their product is better. While I was going through the literature said more the components they accumulate in one tablet, the efficacy of the products goes down. So if you just see, if there is a zinc which is 50 milligram, it is effective at this dose. This is the minimal effective dose. So if you're combining all these products, you cannot have efficacy of all the products combined in one tablet. When all the tablets are combined, definitely they tend to interact with each other, and then the efficacy of the antioxidants also goes down. And for how long, Dr. Shweta, if at all you have to recommend, then for how long do you recommend?
Yeah, so I still follow the old, thought process that one whole sperm cycle is 3 months. So if you have to give, but the thing is to be thought is that all these antioxidants will actually work at the storage level, not at the testicular level. They can only work at the epididymis because other places is the blood testis barrier. So possibly giving it for 2 months is something which I'm looking at.
Yes.
So when you read the literature, then you have data for 3 months, 6 months, and even up to 1 year.
True. And Dr. Vineet, coming to you now, though this has already been said by Dr. KD and Dr. Shweta, what is your preference of giving antioxidants? Suppose you're working in a periphery where the couple doesn't, I mean, the patient cannot afford such expensive antioxidants. So given choice, what would you recommend?
I think it's already answered.
Yeah.
I give B complex with C. That's with zinc. So that's the easiest thing to give at the lowest cost possible.
Yeah, very true. And all these antioxidants, they basically the role of the antioxidants that they act as scavengers. Now, whether you give lycopene, whether you give OQ, whether you give carnitine, the ultimate goal is that they act as scavengers, and which can be fulfilled by giving these vitamins and folic acid and zinc. And Coming to vitamin D, Dr. Vineet, what's your take on this? Do you give it to all the patients? Because these days,
No,
at least in female infertility, we see a lot of prescriptions coming with us. There has been, a trend to write so much vitamin D. So what about the male
definitely not.
infertility?
No,
Definitely
definitely not.
not.
I don't think there is evidence to show that vitamin D deficiency has any role in male subfertility. And you may correlate it to a lot of abnormalities in every system of the body, but you'd have to prove severe persistent deficiency to show associations.
Okay. Anyone giving vitamin D? Dr. Shweta, Dr. Sujatha, Dr. Pranay, Teena, all my panelists.
No,
Is
ma'am,
anyone
routinely, we don't routinely give vitamin D.
Yes. Dr.
Yeah.
Shweta?
Yeah, no, I don't give vitamin D at all to male partners. I hardly remember giving it to anyone.
What about the female partners? Are you giving it to the
Very
female
little.
partners?
No, not for the infertility workup. I'm not doing it.
Okay, great. So the take-home message is that vitamin D is not to be given routinely.
But as a
And
part of, as a part of infertility workup, we would do the vitamin D levels. I don't know if all other panelists do it or not, but we, especially for the female partner, we do the vitamin D levels, and we prescribe a vitamin D only if the levels are suboptimal.
We are not
And
doing
the
vitamin D
testing,
levels.
again, Pranay, the
female.
testing is more expensive than the.
Exactly. So we are
So
also not doing
current-
vitamin D levels.
So none of the current guidelines mention vitamin D estimation as a assessment tool for male subfertility. And we can look at Indian guidelines, we can look at the European guidelines, or even the ASRM or the AUA guidelines. None of them mention vitamin D estimation as a test for subfertility in the males.
Yeah. So this question goes to Sujatha and Pranay. How do antioxidants impact pregnancy rate and live birth rates? Sujatha?
So I think the data is not very conclusive on that because first of all, like how long to give antioxidant itself is not very clear because, like this 3 months does not seem to have any impact on the pregnancy rate is what many publications they say. So to the best of my knowledge, like there is no very strong evidence on antioxidants improving pregnancy rates or live birth rate.
Pranay?
Yeah, ma'am has worded out eloquently. The Cochrane 22 shows that perhaps there is some evidence of benefit with regards to the pregnancy and live birth rates, but it is coming from one of the 17 studies, and that skews the whole measure. So
there's only one study in the whole Cochrane which says that, right?
One out of the 17 that reports on live birth rates out of the 17. And then again, in the recommendations, they say that this should be taken with a high level of bias in reporting. And that, of course, as Cochrane says, with all other studies, more studies are required. Then there was a recent June 2023 meta-analysis by Dr. Ashok Agarwal. They did show that there is some improvement in the early clinical pregnancy rates, but not in the live birth rates. But there's so much heterogeneity. You don't really know which thing is working, what add-ons are working. It's not the only thing that you're trying for patients who are trying to conceive with male factors of fertility. Apart from antioxidants, they are being subjected to ICSI, they have been subjected to sperm sorting. So you really cannot say. So for you to have an adequate measure of effectiveness, you would have to randomize patients who are solely given antioxidants and subjected to either conventional ICSI with nothing else, and have the other subgroup without antioxidants. So that's not happening. So it's really not easy to say what's working or what's not.
Not a feasible this thing. Shalini, go ahead. There are studies which are showing an improved rate, but those are poorly done studies. Like you said, there's so much heterogeneity.
Yeah, Dr. Devanshi, after hearing so much, like everybody said that they give, if at all required, the antioxidants for 2 to 3 months. But yes, we do see prescriptions in our OPD where the patients are given antioxidants for long and long, maybe 6 months. It is being continued. Patient moves from one center to another. Everybody writes their own favorite antioxidants. So do you feel that they are double-edged sword in the sense that if given long, they have a harmful effect?
Ma'am, reactive oxygen species have dual role in male infertility. Because low levels of reactive oxygen species are actually needed for capacitation, hyperactivation, and normal sperm function. But if we give too much antioxidant, then there is reductive stress, like Pranay sir has said. So that may in fact hamper, and ROS will not be sufficient to maintain the normal sperm function also. So we have to keep in mind to not tip that balance towards the reductive side. But at the same time, what we do at our center is that if we find if there's a patient who's smoking or alcoholism history is there, first we tell them to stop that and we put on antioxidants. Then we get a repeat semen sample after 1 and 2 months. So if we see improvement, we continue with that antioxidant. Otherwise, we don't.
Yeah, because I feel giving more than 2 months or almost 3 months, because the balance would shift to a redox balance where
Yes.
they would be more reductive stress rather than the oxidative stress. So this chart very beautifully highlights if the oxidants are more, then it would lead to oxidative stress. There has to be a balance, and if the antioxidants are given prolonged, then it would lead to a reductive stress, which will eventually hamper the physiological actions, and hence again it would lead to impairment in the semen parameters. So, Dr. Surveen, your
Okay, how
question? I think,
raised DFI affecting the fertilization and blastocyst rate? Sujatha, what have you seen in the lab as a practical?
We have seen that there is not much of an impact on fertilization rate, but to a certain extent on blastocyst rate, and that too not on the total blastocyst formation rate, but the usable blastocyst formation. So the good quality blastocyst actually reduces with an increase in DFI. That is my observation.
Pranay, what about you?
We have also had the opportunity to have the patients who have failed to make good quality embryos in the first cycle being subjected to perhaps not if antioxidants, but some sort of sperm sorting. And we have had better blastulation. Literature scrutiny has revealed that, as ma'am has mentioned, that higher oxidative stress, higher sperm DNA fragmentation perhaps does not decrease the fertilization rates. But again, whether it actually decreases the later embryological outcomes like blastulation or implantation rates is also debatable. We feel a little tempted to go along with the flow because we are being subjected to so many sperm sorting devices, a couple of microfluidic devices. There's now another that has been launched recently. So we perhaps take it for granted that sperm DNA fragmentation is affecting the utilizable embryo developmental rate. But there are papers by good groups by the group of Janpiro Pelormo, who have shown that they have drastically reduced the level of sperm DNA fragmentation using microfluidics, and that it is not only translating to better blastulation, but better embryoploidy as well. But then there equal amount of there is equal literature, equal number of publications which is saying the converse, that higher DFI does not alter the fertilization, blastulation, good quality embryo, and the euploidy rate. What is our recent publication, July 23, is showing from China that perhaps it does affect the segmental aneuploidy rate. But this is again just one study that is saying. But the bottom line is we have ample literature for both the arguments that it affects and it does not affect. So you have
practical, what you are doing in your lab.
We are actually using sperm sorting for patients who have failed to give us the stipulated blastulation in the first cycle with decent quality embryos and normozoospermic samples. So these patients are subjected to sperm DNA fragmentation, and we.
What next if the DFI comes raised?
So we have told them that a combination.
Apart from giving antibiotics and diet modification and everything for IVF.
Definitely diet. So we tell them that a combination of shorter abstinence, use of ICSI, and a sperm sorting method is a pragmatic approach to dealing with high DFI. Of course, I know of groups who have published as well wherein even non-monospermic patients, they have subjected the patient to surgical sperm retrieval. But these are again centers, and they have published it, but it is very difficult to counsel and convince patients who on paper have non-monospermic parameters, and they
agree, and you subject them to surgery.
but there have been
So
reports.
I'd just like to make a comment here. We have
Intervention. KD sir, your take on this?
You see, what they say, the DFI sample, DFI sperms may not be affecting the fertilization or blastocyst rate as per the various studies. As a clinician, what I'm more concerned is a DNA, this thing, sperm can cause you, even the patient conceive, it can cause you early pregnancy losses. That is there.
Yeah.
So if you have a sample found with a high DNA fragmentation, probably you have to take some remedial steps, like what you mentioned, short abstinence periods, sperm selection, and ICSI. And one or two patients in our clinic, we have taken their testicular sperm also.
Yeah, so I just wanted to make a comment here. Like Sir said, we did this was a study I presented in ESHRE where we did a short insemination, because prolonged insemination again causes DNA damage. That is what they say. And just like Sujatha said, the short insemination, the fertilization rate was not affected. In fact, it was better with a long insemination. But the blastulation rate was so good. We did a 2-hour insemination and removed the oocyte from the semen preparation. And our blastulation rate was much better, significantly better with a shot. So somewhere ROS is making a difference to the blastocyst rate, definitely. And like Sir said, to the clinical pregnancy rate and early abortions, it is going to make a difference.
So, can I ask one question? Can I ask, ma'am, Sujatha, ma'am, what cutoff value do you follow for this DFI? Like more than 30%? At what value did you observe the less blastocyst rate conversion?
No, anything above 30%, like not in the, see, when it is, like moderate, according to that value, like if it is in between 20 and 30, it doesn't make a significant impact. But another thing which I also have to mention here is the female age also matters. Because if it is just the DFI and a young patient, we have not seen much of a difference. But in elderly patients with high DFI, we see issues with the quality of blastocyst. I wouldn't even say blastocyst rate. That seems to be okay. But the blastocyst which we are freezing or transferring, that
good quality blastocyst.
Yes. Yeah.
Okay. Okay. Thank you.
So Vineet, is surgical retrieval of sperm an answer? Would the testes fare better than the epididymis is the question. What do you feel as an andrologist?
See, routinely, we know that it doesn't make a difference where you take the sperm from. But to answer this question, there have been both sides, again, as most studies that we've seen now, because they're not adequately controlled. Now, when you took testicular sperm, was that the first ICSI cycle? Was that the second ICSI cycle? Did you change the protocol for the ICSI itself? They never measured all those things when they had the outcomes were assessed. And so, because several factors actually contribute to it, and you've modified several of them, you couldn't actually prove that it was just the fact that you took, also the fact that you took testicular sperm in that particular study that you performed, and you got better outcomes. That was the reason for it. Which is why I'm saying we don't have the answer yet. But in a lot of conditions, this does become the next logical step as you would go. You failed 3 cycles, you failed 4 cycles with the ejaculate. What next? You've done everything that you've done, and we've all gone through that process where you've had 4, 5, 6 failed cycles, and you just don't know where and what has gone wrong. You've modified everything possible, and that's the next logical step.
So you were also mentioning ICSI for some patients. What is your experience? Sir, KD sir?
Yeah.
You were mentioning ICSI for some patients. What is your experience on it?
That is what I say. Whenever we have a sample with the DFI, we take cutoff as 25%. We like to do a sperm selection, sperm sorting out, and prefer to do ICSI.
And what about testicular sperm, sir?
I mentioned 2 or 3 patients who had taken up testicular sperm.
Ah.
Those 2 patients, the 3 patients, they had a repeated early pregnancy loss.
So did it
And
help?
We have to analyze our, this thing with the 3 patients is not a big series.
Yeah. Sujatha, what about you? Do you have any?
Sorry, which one? What is that?
Testicular sperm, if you're having repeated failures or abortions with a high DFI.
Actually, like we have not done much of this of late. So only the theoretical thing I would know.
Generally, Dr. Surveen, it is difficult if a patient is able to give semen sample and we tell them, no, your semen sample is useless. We're going to take out surgical, we have to do
Difficult to
surgical.
convince. Yeah.
It's very difficult to convince. Very difficult to convince.
Just like the second ejaculate.
But
it
that's
is
much
more difficult
easier.
second ejaculate.
Yeah. But even that is difficult to convince. You have to sit and tell the patient. So
Mostly this data that of the SSR sperm for SDF, high SDF is coming from centers that are focusing on cell cycles. So the centers that I know of who have published, they're solely focusing on cell cycles because of the religious constraints. Otherwise, I personally have faced a couple of patients who have been counseled that perhaps this is the next best step in view of very high sperm DNA fragmentation in the order of 80 to 90%. And different patients are different. Their thresholds for accepting a sperm donor, which was not even a consideration for me while I was discussing all this. And they themselves mentioned that we will perhaps not want to again do a hit and trial, and we just accept a sperm donor. So it also matters what kind of setting you are in.
yeah. Shalini, is that the last question? Would you wind up?
Yeah. So with this, I think we come to the end of this panel, which is the highlights and the inputs given by all the esteemed panelists. They were great. And I guess the delegates have been benefited by the inputs regarding the DNA fragmentation and the antioxidants, which both are very burning and debatable topics. And I guess if there are any questions, we can take it. Are there any questions in the chat box?
No, not yet.
No.
Yeah, not yet. So I guess with this we wind up, and me and Dr. Surveen sincerely thanks all our esteemed panelists and who have done a wonderful job. And I request Devanshi now give a vote of thanks.
Yes, ma'am. Thank you so much, ma'am. Thank you, Surveen ma'am, Shalini ma'am, and all the panelists. This has been a really engaging and a very informative session. Now, for the vote of thanks, I want to invite Dr. Himanshu Roy, sir.
Dr. Himanshu there?
I'll have to check.
Otherwise
he's
you can.
there.
Okay.
Himanshu, sir?
Okay, Devanshi, you can continue. I think probably is not
Okay,
there.
ma'am.
Yeah.
So just a lot of thank you, ma'am. It has been a really good session. We should have more such sessions in the future. It's been really nice, ma'am. Thank you so much.
Yeah. So sincere thanks to the KD sir, Dr. Surveen. Yeah. Is Dr. Himanshu there?
Yeah, I was there. Actually, it was unmuted for the whole thing, so I could not.
Yeah. Dr. Himanshu, just few words from you.
It was very nice having all of you and speaking about a very relevant subject. Everybody comes across these male partners and using antioxidants. I would just like to give 2 comments. First was regarding this DFI testing and everything. I would just like to share one experience. Like now we are not doing too many egg donations, or very few. But earlier, 3-4 years back, when we were doing egg donation, then one of our students did a study, and it was her PhD thesis on paternal age effect of paternal age on DFI and its effect on the blastocyst rate. So in our study, we could not prove, because in egg donation case, mostly the female factors are controlled. So you only have age above 40 and below 40. So we had 2 arms, and there was no correlation between DNA fragmentation index between these 2 paternal ages, and neither there was any correlation between the DNA fragmentation test and the blastocyst rate. So this was just one of the things, because though there is DNA fragmentation, but it is very difficult to diagnose and treat. So whatever we are doing right now is totally empirical. And second thing, coming again to vitamin D, also just like any other antioxidants that we are using right now, it's true that the role of vitamin D has not been found out. But again, when we studied our patient group, then we found deficiency in 90% of the people coming to our clinic. And so that's why, rather than testing, as Dr. Pranay has said, it is almost, since the prevalence of vitamin D deficiency is 90%, my vitamin D level was 3%. So we give routinely vitamin D to all our patients, both male and female. The effect on fertility is definitely not known, but it is not an expensive drug, and it easily no side effects. And since it has effects on so many things, and especially on bones and maybe muscles, so if it will not help in the directly on sperm quality, it will definitely help in our bone muscular coordination and strength, which will help in natural conception also. So that was one of my takes. So thank you everybody, and the Madhya Pradesh chapter which is hosting it, and the Holistic Society and Special Endocrinology, SIG of the Endocrinology chapter, and Dr. Kuldeep Nair sir, Dr. Surveen. So, and all the panelists were extremely good. So thank you so much for coming to this seminar. Thank you.
Thank you so much, Dr. Himanshu, and special thanks to Atul, who has been there and managing the technical part of it. And IFS team, thanks. Thank you so much, KD sir and Dr. Surveen. Yeah, and Dr. Priya Bhave. I think she is busy, held up in some emergency. So thanks to Dr. Priya also for giving us this opportunity. And looking forward
it's our
for
pleasure
more
also, ma'am. It's our pleasure also to be a part of this SIG group.
Thank you.
So
I mean, we finally calmed down that male infertility, the causes seem to be environmental, lifestyle, and stress, maybe. And the solutions also appear to be holistic. So I think we need to move towards a holistic way of life and treatment, because we're talking of dietary changes and stuff like that, in addition to the drug therapy, which is also a little empirical. So thank you all of you for participating and joining SIG. Thanks to all the panelists also.
Thank you so much.
Oh, yeah.
Thank you.
With this, we all take a leave.
Bye.
Bye-bye.
Yeah,
Thank you.
bye.
Thank you, Dr.
Bye.
Shalini.
Bye.
Bye.
Okay, thank you, ma'am.
Thank you.
Bye. Thanks, everyone.
Bye.